So the idea was that in looking at thin sections of yeast cells, one can see some organelles that look like they could be associated with secretion, not as dramatic as in a section of mammalian cells. But Novick and I guessed that the vesicles that one sees congregating under the bud portion of a dividing cell would be the vehicle not only for secretion but for assembly of the plasma membrane. And so we predicted that anything that blocked the biogenesis of these vesicles would be lethal, and therefore one would have to go after conditional temperature-sensitive lethal mutations. So that was the first and crucial assumption, that it was going to be an essential gene. And the second was that the mutant would, at a restrictive temperature, cause secretory proteins, which are otherwise very efficiently secreted from cells, to be accumulated inside of the cell. And that actually is a really trivial assay. You just assay intact cells, which measures the enzyme that's been trapped in the cell wall, because the cell wall is porous. And then you dissolve the cell wall with an enzyme, and you have spheroplasts that are left, and you just rupture the spheroplast with a detergent, and you assay what's inside the spheroplast. And that's a measure of accumulation. In a wild-type cell, that baseline is virtually zero. In a sec mutant, it rises dramatically above the background. So the first mutant, just based on a biochemical enzyme assay, was really obviously blocked in multiple secretory enzyme transport. But the dramatic result came just around the time George Palade visited Berkeley for a special lecture. And I recall Novick telling me that at a luncheon, Palade had encouraged him with these preliminary results to cut a thin section and look and see what these cells look like. And so we had an expert technical assistant in the department. And that, of course, was one of the great eureka moments of my career when Novick excitedly called me down to the EM room and showed me this Sec1 cell sections that were just, like they had measles. They were just full of vesicles. And nothing like that had ever, at least never been published. I don't think it had ever been seen before. And from that moment, really literally from that moment on, I knew that we would have at least several decades worth of productive work.